Alrighty, here we go with the first set of notes in our genetics series. This one we're calling Intro to Genetics. In this set of notes we'll lay out a lot of the terminology that we're going to talk about in the future. A lot of it should probably seem familiar to you. You've been exposed to a lot of it before. Some of it might be a little bit new or a little bit hazy, so we're just going to review some of that terminology. To begin with, let's do a little bit of the history of genetics. The man who's considered the father of genetics is this guy here named Gregor Mendel. He was a monk living in Germany and working really pretty early in the 1830s, 1840s, and did some pretty cool stuff. He used pea plants to research heredity. Genetics is basically the study of heredity, and heredity is just the passing of characteristics from parents to offspring. Right? So if you think of something that's hereditary, maybe a disease might be hereditary. It's because it's involved with your genetics. It's in your genes. Gregor Mendel worked with pea plants. He worked with pea plants for lots of reasons. One, they had these 7 traits that were really, really easy to see. So he didn't need any special fancy scientific equipment. He could easily see if a seed was round or if it was wrinkled, if it was yellow or green, if the pods were wrinkled or smooth, if they were yellow or green, where the flowers were the flowers on the end, terminal, or were they all along the stem, axial, or is it a tall plant, or was it a short plant? These are really, really easy things to see, no specialized equipment needed. It was also really, really easy to breed plants. He could say, hey, I want this plant with yellow pods to mate with this plant with green pods, and he could make that happen. And they grew up pretty quickly. You know, he could do 2 or 3 generations of something right there in his garden. He didn't have to have cages full of monkeys that took a couple years to mature and things like that. This is something that was really, really easy. And when he was done, they probably got to eat what was left over, so that's also a nice little bonus. One of Gregor Mendel's greatest contributions to genetics was figuring out the whole dominant and recessive concept. He figured out that traits, or things that you see, the characteristics, he figured out that traits were controlled by 2 alleles. Now, he didn't call them alleles. He didn't know what the heck a gene or an allele was because we hadn't really even discovered DNA at that point. He called them factors. All right? And so basically he took some plants and he bred them. Right? So here he has the green plant and a yellow plant. This is our parental generation. And he had them breed together, and then he got a whole bunch of plants that were green. In the first generation of offspring, we call this the F1 generation. So they were all green plants. All of them have green pods. So we said, oh, okay, so green must be, quote-unquote, better than yellow. Well, I wonder what happens if I breed these guys together. All right, so we took a green plant and bred it with a green plant. Probably expected to get all green plants again, but if we look here, that's not what he got. He got mostly green plants, 3/4 of them, 75% were green, but he also got some yellow plants in that F2, the second offspring generation. So he said, what the heck? How did I get a yellow plant when I was breeding green plants? That doesn't make any sense. So the only explanation was that there were 2 factors that were controlling the color, and one of them was hidden in here. So these guys actually have 2 factors. We're just not seeing one of them. And that's where you get the whole dominant and recessive thing. So you can have some traits that are dominant, so the trait that gets expressed or seen. And whenever we're writing this, we always write this with a capital letter. Or a trait could be recessive, so the trait could be hidden by the dominant trait. And whenever we write these, we would write these with a lowercase letter. So like I said, he didn't really know anything about genes or alleles. He just called them factors, but we do know stuff about genes and alleles. We know that a gene is just a length of DNA on a chromosome that controls a specific trait. So remember, we've got all of our chromosomes come in homologous pairs. If we're talking about humans, we have 46 chromosomes and 23 homologous pairs. Right? You get 1 chromosome from your mom, 1 chromosome from your dad. They're the same thing. And so in this example here, we've got a chromosome from a plant. And here at the end of the chromosome, on both of them, notice in the same spot, we've got the gene for flower color. All right, so that's this bit of DNA on all of these types of plants are always going to have that gene in the same spot that controls the color of the flowers. Okay, but sometimes those genes are a little bit different. They send a slightly different message. That's the allele, or the factor that he called them. It's one of the alternate forms of the gene on a homologous pair. So that's where the dominant or recessive comes from. Right? This gene is saying we're going to make purple flowers, And this gene is saying, no, we're going to make white flowers. So they're both genes for flower color, they just have a slightly different message. And one of them is going to win out. Now, if purple is dominant, then the flowers would have, or the plants would have purple flowers, or vice versa. So the product of all Mendel's work were these couple laws that he came up with, which are actually things that we already know about because we know stuff about meiosis, which he didn't really know about. The first one was the law of segregation, that alleles are segregated or separated during meiosis. Of course, he didn't know what the heck meiosis was, but we already know this, right? When we do meiosis, we know that we start off with a diploid cell that has 2 copies of every chromosome, and then at the end of meiosis, we have more cells that have only 1 copy. Those 2 copies got separated into different cells. That's segregation. And he also figured out the law of independent assortment as well, that his alleles or his factors are separated randomly. And we learned about that in the last unit as well, how the mom and dad chromosomes, they don't always stay on one side of the cell or the other. They mix randomly so that the offspring are a random mix of all of your chromosomes. I think it's kind of neat that he figured all this stuff out without really modern scientific equipment or any genetic equipment or anything like that. He sort of figured this out way before we knew about mitosis and meiosis. So moving on to some terminology that we're going to use a lot, that is homozygous versus heterozygous. We've seen words that start with the prefix homo before. We've got homeostasis, which is the same state. We've got homologous, which is the same chromosomes. All right, so in this case, homozygous, in this case, both of the alleles in a pair are exactly the same. So you're getting the same message. So homo means the same, so in this case the alleles are the same. And you have 2 different options for homozygous. You could be homozygous dominant, so you would have big T, or you could be homozygous recessive, little t. Right? Both of the alleles are the same, they're the same letter, big T, or little t. And then your other option is heterozygous. Remember, hetero means different. And so now you have 1 dominant allele and 1 recessive allele. So hetero means different, alleles are different. So heterozygous would be like a big T and a little t. So you have different letters, one's big, one's small. That's heterozygous. Really, really important terms. We're going to use these a lot. When you say heterozygous, you don't have to say heterozygous dominant. We know that it's dominant because we know there's a dominant one in there. But when you do say homozygous, you do need to be more specific because there's 2 different options. You can't just say, oh, that person's homozygous for that trait. Well, that's different. Homozygous dominant is going to produce a very, very different result from homozygous recessive. The last bit of terminology for this set of notes is genotype versus phenotype. The genotype is just like it sounds, it's the genetic makeup of an individual. I like to say it's the type of gene. Okay, so this is the letters that we're actually going to write to represent the genes. It's really important that we always use the same letter for a given trait, and that'll become a little bit more obvious what I mean by that in just a second. Versus phenotype is what you actually see. The phenotype is the appearance of an individual. The phenotype is controlled by the genotype, so based on what kind of genes you have, that decides if you're going to be tall or short or whatever. And it comes from the Greek, pheno means to show, so this is what actually shows through. So if we look over here at this example, we've got a phenotype of a tall individual, and we see that his genotype is homozygous dominant, so the tall is obviously the dominant trait. So if tall is the dominant trait, and we come down here to our heterozygous individual, he has a big T and a little t, so what must his phenotype be? Well, it has the dominant allele, and we know that tall is dominant, so this phenotype is tall. So then we move down to the next line, and we've got a short individual. And I told you a short, what is his or her genotype? Well, the only way you could get a short genotype, if tall is the dominant, then it would be little t, little t. And this is where it becomes really important to always use the same letter. Sometimes people get confused and they would say little s, because they think, oh, short starts with an S, I'll use an S. No. You can't do that. It always has to be the same letter. If we're talking about the gene for height, we're going to use T in this case. Usually, the letter you use is the first letter of the dominant phenotype, so T for tall. If we were looking at another characteristic, like maybe fur color in rabbits, and we said that black was dominant over white, then we would say usually a B letter for that. So there we go. That wraps up the first set of notes that introduce you a little bit to some of the terminology of the genetics unit.